seen the reactivation of this silent locus. Now, it's not to say that if one didn't use some alternative strategy, one couldn't reactivate the locus. But using current methodology for iPS-based reprogramming, this locus, almost alone in the genome, remains resistant to the reprogramming process. So it must be densely heterochromatic, and indeed we show it's methylated and carries silencing histone modification, and that remains resistant to the global internet modeling that otherwise goes on with reprogramming. And so here's an example where if your interest is to study the mechanisms of the dynamic transcriptional silencing that occurs at this locus during human development, one should use the embryonic stem cell model because the iPS cell model won't reactivate the locus. So another example of a way of using pluripotent cells to model disease is the resource of preimplantation genetic diagnosis, which generates embryos that would be discarded as medical waste. But through work primarily done at the Reproductive Genetics Institute in Chicago, there are now dozens of different genetic conditions that have been reduced to embryonic stem cells. Now, unfortunately, because of problems with the informed consent that was used for all of these many cell lines, they've not received approval under the new NIH policy. And so what we're hoping is that more groups will step forward with properly constructed consent. And indeed, we've begun at Children's Hospital, in just a small way, we've now isolated 3 PGD-DS lines from the conditions listed here, and we hope that that number will grow over time. And I think we're going to find examples where having both the embryonic stem cell side by side with the iPS cell is going to allow us new insights into disease. So let me summarize the first part of the talk, that iPS cells do enable a wide array of in vitro models, and currently they're most useful as tools for research and of finding their way into the drug discovery pipeline. We have many, many issues that remain to be faced before we can be comfortable that these cells will be ready for any kind of cellular therapy. We appreciate that it requires a very prolonged culture to generate these cells and characterize them. And there's emerging evidence that this prolonged period of culture can lead to genetic as well as epigenetic instability. We have to refine methods for gene repair so that we can exploit. And ultimately, we need to get better at the directed differentiation because the production of cell types that could be effective in a transplantation model remains once again a challenge. I think I've pointed out that there are going to be diseases that are not amenable to modeling with iPS cells and for which embryonic stem cells or other traditional models may be more effective. And I want to leave you with the notion that embryonic stem cells remain essential tools for research, alternatives in some cases and perhaps preferential sources in others. And in the second half of my talk, I'm going to bring us back to this notion that nuclear transfer as an approach to reprogramming may yet teach us important lessons that will improve our ability to generate iPS. So the question that I'll spend the remaining amount of time on is this important one, for determining whether iPS cells are really equal to iPS cells. We know that reprogramming creates a generic iPS cell, which in the optimal circumstances, when one starts from mouse embryo fibroblasts, when one can select for reporter genes, when one can characterize the cells that practices complementation and germline transfer, that the generic ideal iPS cell is in fact functionally and molecularly indistinguishable from embryonic stem cells. But in practice, it's actually quite different. And we've recently published, as well as Alex Meissner and Catherine Platt's lab, that you can get frozen in intermediate states along the reprogramming process. and that these intermediate states can look very similar and even function to some degree as pluripotent cells. And that under some circumstances, one can convert these intermediate states to a fully reprogrammed state by interventions that minimize residual demethylation. And what's been emerging in the literature is the notion that in various isolates of iPS cells, there does tend to be a residual measure of gene expression from the donor tissue. And this is something that we hypothesize in practice really represents a memory, an epigenetic memory, something superposed on the DNA that is a memory of the tissue of organs. So I'm going to discuss how we've explored this issue. And it starts with a very close collaboration between my lab and that of Andy Feinberg. And Andy's group, together with Rafa Irizarry at Hopkins, has pioneered the development of an array-based method to assess methylation across the genome. They call this method CHARM. And on the array is represented 4.6 million CpG sites. So it's comprehensive methylation across the genome. But it's importantly missing the non-CpG methylation. And by comparing differences between fibroblasts and iPS cells, we've been able to identify a large set of differentially methylated regions, which we call reprogramming DMRs, reprogramming associated differentially methylated regions. Here's just one such region in the goosepoid locus, where you see in brown is the parenchymal fibroblast, and in red is the mean measure of methylation in the iPS locus. And you see this region is affected by demethylation during reprogramming. And one can take this array-based method and then independently validate these loci using pyrosequencing. So here you see just across this 9 different iPS cells as well as 3 ES cells that the locus is demethylated in the pluripotent state and it's methylated in the fibroblast. So if one uses these differentially methylated regions, the reprogramming differentially methylated regions, you can actually cluster different tissues. So these reprogramming DMRs will faithfully separate liver from spleen from brain, which tells us that those regions, even in the fibroblasts, that are reset during the return to pluripotency, are those regions that are intrinsic to cellular and tissue identity. Now what we then focused on were not the 98% of those regions that get faithfully reset, but we focused on the 2% that were consistently different between ES and iPS cells. And indeed, this is just one such region here in this phosphatase locus that by pyrosequencing across these 9 iPS cells remains methylated in the iPS, but is not then reset with ES-like state. Now this was a curiosity. In some loci appear to not be reset to their ES-like state. We published this in Nature Genetics. It essentially suggests that there are differences between iPS and ES cells where there's a tendency towards residual hypermethylation at these loci. And this has been independently confirmed from Kun Zhang's group at UCSD. So the question is, what's the significance, if any, of these few residual regions of hypermethylation? So we decided, instead of trying to ask this question in the more genetically complex human cells, we returned to the mouse. And this is the work of Kitwa Ng, working with a postdoc, Kitai Kim, in my group. And we're asking for a very comprehensive functional and molecular comparison of a variety of different pluripotent stem cell types. And I should mention with great gratefulness
Today's world speaker, Dr. Karen Gass. She's a professor of pathology and cell biology at Columbia.